Well, good afternoon, everyone. Welcome. We're going to wait a couple minutes to get started so everyone can log on. If you're just joining us, I want to say welcome this afternoon. We're still waiting for all of our participants to get logged on, so we'll be starting momentarily. Okay, ladies and gentlemen, I have 12:01 on my end, so out of respect for everyone's time, we're going to go ahead and get started here today. Again, good afternoon. My name is Jennifer Chrisman. I'm the Vice President of Watershed Conservation here at the Western Pennsylvania Conservancy. Thank you for joining our Earth Day webinar today entitled Meaningful Mussels, an overview of freshwater mussels in Pennsylvania and a look behind the scenes at some of our work to conserve these animals and their habitats. We have more than 100 people registered here today. If you're a WPC member, I want to thank you very much for your membership and thank you for your support. If you're joining us for the first time today, welcome. Western Pennsylvania Conservancy is a nonprofit conservation organization founded in 1932. This is our 90th year of working to protect and restore our region's most exceptional places. Our work includes protecting land and wildlife habitats, restoring and reconnecting rivers and streams, providing nature preserves for the public's enjoyment, studying threatened and endangered species and their habitats, and greening communities with trees, gardens, and green spaces. And also, last but not least, protecting Frank Lloyd Wright's Falling Water. The very first Earth Day was in 1970. By the end of 1970, that first Earth Day had led to the creation of the U.S. Environmental Protection Agency and the passage of the first of their kind environmental laws, including the Clean Air Act, then the Clean Water Act, and after that, the Endangered Species Act. What our staff will share with you here today are not only exciting discoveries of science, but show that these animals now are found in and in some places thrive in the rivers of Western Pennsylvania. This means that those federal laws made possible by that first Earth Day and the work of WPC and our conservation partners are in fact making a difference to improve the water quality of our region. Our speakers here today are Eric Chapman and Mary Walsh. Eric Chapman is WPC's Senior Director of Aquatic Science. He's been part of our watershed conservation team since 2006 and oversees all scientific data collection and monitoring related to eastern hellbenders, brook trout, as well as freshwater mussels. Mary Walsh coordinates the Aquatic Zoology program for the Pennsylvania Natural Heritage Program. She's been studying aquatic fauna with the Heritage Program since 2003. Mary works on inventories of aquatic invertebrates and communities, and she models species distributions and tracks invasive species. If you have any questions today during our presentation, please place those questions or other comments in the chat feature. We'll be monitoring that chat and sharing questions at the end of the presentation. Again, I thank you so much for joining us. Happy Earth Day, and here's Mary.
Great. Hi, I'm excited to be here talking about one of my favorite groups of organisms, freshwater mussels. And Eric and I are going to be kind of passing the baton back and forth during this presentation and talking about these topics here. So we'll tell you just a bit about, what the heck are freshwater mussels, how are they different than other kinds of mussels you might know, what's their life history, some other just kind of fun information about them. We'll give you a tour of some of the Pennsylvania watersheds and the work that we've done to study rare species. We'll tell you about a big project we worked on in the Allegheny River to move a lot of mussels from an area called Hunter Station, and how that led to some other interesting questions, and an experiment that we conducted last summer in Western Pennsylvania to figure out where we might want to move mussels to restore them in streams. And then we'll leave you with some messages about some ways that you can volunteer or help out related to freshwater mussels and other programs at WPC. So next slide, please. Okay, so let's start from the beginning. What are the different types of mussels that you may have heard of? Most people's association with mussels are the ones that you've eaten, like the saltwater blue mussels, which are a delicious edible mussel in that right upper picture. And then, of course, in Pennsylvania, we have zebra mussels, and now a sister species called the quagga mussel, which started out invading Lake Erie 30 some odd years ago, and has now spread to other watersheds in the state. And so those mussels have gotten a fair bit of attention in news articles. And the zebra mussels are generally, so those are on the far left side of your screen there. They're small mussels, 1 to 2 inches, and they attach to rocks using byssal threads. And so you'll see them in colonies and marinas and intake pipes. And if you have a boat in Lake Erie for a long period of time, you might get some zebra mussels on the hull. But we're not talking about those either for this presentation. We're talking about our native freshwater mussels that are their own group of organisms. And so for the most part, they don't have those byssal threads that they used to attach to things. In fact, they live in the rocks, in the mud, in the gravel, at the bottom of our rivers and lakes. And so they can be, a couple inches long to maybe the size of the palm of your hand. There's over 1,000 species worldwide. In Pennsylvania, we have records for 67 of those species. So we'll touch on a few of those today. Next slide, please. So a lot of people want to know, well, can you eat them? That's one of the most common questions we get when interacting with the public about these really cool organisms. So the short answer is no. Technically they can be consumed, and they were an important food source to Native Americans. However, because they're filter feeders, they may have contaminants like heavy metals. A colleague of mine did try eating them, and she described the texture as being like eating a rubber band. So I probably wouldn't recommend it. Native Americans did use the shells for tools. They can be shaped into different functional tools, and people have collected freshwater mussels for pearls. There was a big pearl rush in the 1800s. They do produce pearls sometimes. I think it was a popular idea to collect them as jewelry and, something that can make people money. But a lot of freshwater mussel beds, got destroyed because people thought they could get rich quick finding a pearl. And then people have used them to make pearl buttons. The inside of the shell is a real shiny iridescent surface, and so it's very attractive material, and it was used for making buttons at one point. So next slide, please. So we get to the part about freshwater mussels that we kind of have to talk about in these presentations, which is unfortunately that a lot of freshwater mussel species are declining, and that's been long recognized worldwide and in the US. The figure on the left is from a 1997 report that shows 70% of the US species were vulnerable, imperiled, or critically imperiled. And so that trend continues in the Pennsylvania State Wildlife Action Plan. 80% of our mussel species are considered species of greatest conservation need, meaning they're in need of conservation actions by our state management agencies. 14 of our species in Pennsylvania are considered extirpated, meaning we haven't found them in the state for a long time, and we think that they no longer occur here anymore. And we do have a number of endangered and threatened species, indicating, that they're pretty rare, and that, their habitats are important for protection. Next slide, please. So, the reason that our mussel species aren't doing well, that comes from kind of a long history of pollution and habitat degradation. In Western Pennsylvania, we've had a long history of manufacturing and resource extraction, which of course has consequences for our water quality. Coal mining and deforestation certainly were really rampant in Western Pennsylvania, and during the beginning of the Industrial Revolution. We had other industries like tanneries, which were particularly polluting, and the Clarion River once was the color of just black water because of the pollution from that industry. Of course, later steel manufacturing and other industries made creeks uninhabitable. Some of the legislation that Jennifer mentioned at the beginning of the Clean Water Act has really helped improve our water quality. And so in places that, our pollution has abated somewhat, but dams are also very widespread, and they're very habitat altering. So they change a system from a flowing water to more of a lake-like environment. So very different. And they create large areas of sedimentation behind dams. I mentioned zebra mussels a minute ago, and they have been, very devastating to some of our freshwater mussel populations. They do kill the freshwater mussels by colonizing their shell and just, making their life pretty miserable and sometimes causing death. And round goby is another species that has invaded some of our waterways, and they are a species of fish that eat native mussels, so we are concerned about those. So now I'm going to pass it off to Eric for this next section.
Thanks, Mary. So we've learned a lot about mussels so far, but one of the things that we come across all the time when we're out surveying is, who cares? Why are you guys doing this? Why are mussels significant? Mussels are significant because they're such a long-lived group of animals. Freshwater mussels are invertebrates, so they have no backbone. But the fact that they can live a long time is really surprising. Most invertebrates don't live very long, 3 years, 5 years. Mussels are different. They are extremely long-lived, and we have some species that can live up to 100 years. So it's really surprising that they're able to do that, especially given the fact, like Mary had touched upon, that they're filter feeders. So if mussels live their entire lives in the water column, Charles Beer, our Senior Director of Conservation Science, has coined the term, mussels are the canary in the coal mine for our streams and rivers. They are going to be the first group of animals that are going to disappear if the water quality conditions become poor. When those mussels do die, they leave behind historical data in the form of their shells. We are able to use shells when we're out doing surveys from animals that that we find when we're out in the rivers and streams. We also go down to the Carnegie Museum of Natural History, where we can look at shells that are over 100 years old and look at the type localities and kind of get an idea of what the fauna was before a stream became polluted. Probably the most fascinating aspect of freshwater mussels is their reproductive strategy. Freshwater mussels, need a fish host to complete their life cycle. It's fascinating. And there's one group that's taken, one species is taking it even further than that. The salamander mussel, Simpsonias ambigua, actually needs a mud puppy to complete its life cycle. So the fact that mussels live 100% in the water, they filter feed, they need a fish host, sometimes species-specific fish host, they have become highly endangered. And that figure that Mary showed you really harps on, how endangered they're becoming. This picture here that I have on the screen, if you can see my mouse moving around right there, that's a mussel right there if you were looking through one of our mussel buckets. So you guys are out on a survey with us right now. So here's the life cycle of a freshwater mussel. The males release sperm into the water column. The females are filter feeding all the time. That's what mussels do to make their living. So she brings the sperm in, fertilization happens internally. The most fascinating part about all of the reproductive point of this is that a mussel that has absolutely no eyes has never been able to see before, but it is able to modify its mantle flap to look like a fish. Some species can modify them to look like a crayfish, complete with an eye spot. It's wonderful. A bass, a sunfish, a rock bass will come down and try and bite that lure, and the female will shoot those baby clams, little glochidia, they have little hooks on them, into the mouth of the fish. Those glochidia will then hook on the mouth, they'll hook on the gills, some will hook on the pectoral fins. The fish swims off and goes, wow, I thought I was going to eat and I got something in my face and that's not cool. And lo and behold, that fish moves upstream. The juveniles hang on, the glochidia will hang on there for a couple weeks to a month. And then when they're fully metamorphosed, they drop off as a free living mussel. That's how mussels are able to disperse upstream. They're using fish to be able to disperse upriver or downriver. So Alicia Trexler is really good at finding things when we're out doing our surveys. And this is one of her pictures. And you can see this is a female displaying her lure, complete with the fins here. And you can see an eye spot. This is one of my favorite pictures. We didn't take this, but you can see these beautiful darters in the foreground here. And this is a mussel, and there is her lure, complete with the beautiful eye spot, fins to boot. Absolutely fascinating that an animal that has no eyes is able to do this. It just blows my mind. So to take it one step further, this is out, all of us, Mary and I, out surveying in Tynesta, and Alicia has the camera, and we came upon a female, and she wasn't doing her thing, and Alicia's like, I got to get a picture of this, I got to view this. So we took some video, and this female thinks that Alicia is a largemouth bass, and she starts doing her thing, and she starts waving her lure, and you can see all the glochidia in these little chambers within inside her gill. Fascinating to be able to see that when you're out doing surveys on the stream, and to be able to share that with you guys right now. It's very cool. So you're getting the bird's eye view of a female mussel displaying. Well, some mussels have to take it to the extreme, and the snuff box is probably the best example of that. The snuff box has modified its shell so that the female has hooks on the shell that she can actually grab a hold of a fish and pump it full of glochidia. So instead of just waving a lure, she's going to actively grab a hold of the fish. You'll see my favorite species of fish, the log perch, swimming up here and rolling little pebbles and cobbles looking for food. Well, this poor unsuspecting log perch gets more than it expected for, and gets grabbed on by that female snuff box, and she just holds on to it until she's ready to start releasing glochidia. So this next little video is going to show you how she's actually pumping glochidia into this poor log perch. And you can see what looks like snow right here. Those are all little tiny baby glochidia that are getting actively pumped into this log perch. Who knew mussels could be so aggressive? So mussels are challenging to identify. Mary and I often have really interesting discussions. We're out in the field trying to figure out, what is this species? You use key shell characteristics on the outside of the shell with tubercles and pustules and all kinds of different things. There's wonderful, beautiful markings on some species, some don't. They're extremely variable across species. There's wavy rays, there's blotched rays, there's chevrons. But the inside of the shell is what we really want to see, because that makes it easier to determine what species you have. But in order to do that, you would have to kill the mussel, so we don't typically do that. We rely on some of those shells that we'll find out on the landscape using lateral teeth, the beak cavity, and then pseudocardinal teeth to identify. Mary and I have been doing this for over 20 years each. A lot of our partners have been doing this longer than that. Using those characteristics like the color, like the shape. In this picture right here, we were up in Tionesta Creek last year, and you have 4 agencies here looking at mussels on this table, trying to identify a particularly problematic species in Tionesta Creek. Mary and I have wrestled with this species many times up in Tionesta to try and figure out what these are. And it's important that you get the right identification on a mussel, in this case, because it's potentially going to be listed on a federal status. So you need to make sure that your identifications are correct. And mussels can be a pretty scary taxa for new collectors. So Mary's going to take it back over now and give you on a tour of Western Pennsylvania watersheds.
Great. So yeah, well, I'll tell you a bit more about some of the information that we've gathered in these watersheds, and why we need to know. Well, that's because the management of freshwater mussels relies on our knowledge of where they occur, and our laws that protect rare species habitat really only focus on the actual locations of where they're located. So nowadays we have a fair amount of information about freshwater mussel distributions in the state, but it's taken a long time to get here. And WPC has played a big role in that, in part because of our participation in the Pennsylvania Natural Heritage Program. Next slide, please. So the Pennsylvania Natural Heritage Program, PNHP, is a partnership between Western Pennsylvania Conservancy, Pennsylvania Game Commission, Pennsylvania Fish and Boat Commission, and DCNR. Its mission is to provide scientific information and expertise to support the conservation of biological diversity in the state. And the PNHP component hosted at WPC has a responsibility for inventorying and monitoring rare species, including endangered and threatened species, and maintaining that information in a statewide database that has a lot of applications, including environmental review. And so when PNHP started in 1982, data about freshwater mussels were pretty spotty at the time. And largely, we didn't have a lot of good information about what species were rare, what were their habitats. That information we take for granted now. Charles Beer, our Senior Director of Conservation Science, is pictured here on the slide. In one of the early, some of the early days of our program at WPC, we started exactly 40 years ago in 1982. And so he began tackling this big task of finding out what mussel species are rare and where they occur. So the data from our projects that we've completed has a lot of applications, like conservation ranking. Our global and subnational ranks get used in a lot of analyses and to help prioritize species for different things. We also roll up our information from our surveys into conservation-based biodiversity analyses, and we designate areas as natural heritage areas when they are particularly special for their biodiversity and rarity of organisms that occur there. A lot of the information that we gather in our mussel surveys gets used for the analyses that go into endangered and threatened listings. We've done some things like modeling habitats to understand the relationship between habitats and the watersheds that mussels occur in, and helped use that information to direct on the ground conservation. So next slide, please. So in the next few slides, I'll walk you through a few projects in each of the major watersheds in Pennsylvania. The Natural Heritage Program has systematically been collecting information on these places. And so we've done surveys of freshwater mussels in the Delaware Basin, the Susquehanna and Potomac Basin, the Lake Erie and the Ohio watersheds. So in the Delaware Basin, we've studied small stream species like the eastern pearl shell. That's that black, really dark mussel, and it's distinguished by that brown foot that you can see in that picture there. So that's unique to that species. It does occur in trout streams, which is sort of unusual for freshwater mussels. It likes streams that are more acidic and cool. it lives 100 years or more, or at least it can. And so we studied the population sizes and we tried to figure out where it occurs in that part of the state. And we worked with the College of New Jersey to analyze the genetics of those populations. We also studied some large river species like the dwarf wedge mussel. So that's a federally listed species. Its habitat in the Delaware River is managed in part by the water that comes out of the dams in the major tributaries to the Delaware, so which is also where New York City gets its drinking water. So there's an interesting, some interesting management implications to knowing about dwarf wedge mussel and how the water there gets managed. Another interesting one we studied was the tidewater mucket, which occurs in the tidal freshwater part of the Delaware River. So the tide in the Delaware estuary pushes and pulls on the fresh water in the lower part of the river where this mussel occurs. And there's kind of a unique set of fauna that live in that environment. So we worked with the Partnership for the Delaware Estuary and the Academy of Natural Sciences to find out some more information about the species. And the results from our partnership and our study was used to assess the species for a state listing. Next slide, please. Okay, we also studied a number of species in the Susquehanna and the Potomac basins. Some of our species like the eastern brook floater and the eastern lamp mussel are relatively rare in our watersheds in the center part of the state, and they may be considered for state listings in the future. Another species, the yellow lamp mussel, seems to have a stronghold in Pennsylvania, and its range seems to be expanding. It's a large river species, and it's occurring in a lot more places than we knew it was before. So that one we think is doing relatively well in Pennsylvania. Next slide, please. Okay, so moving to the western part of the state, these watersheds have a lot more diversity than our eastern watersheds. The Ohio Basin, for instance, has 54 species. Of course, it does have a history of severe pollution, but we think it's recovering a lot of places. And the Heritage Program has worked in concert with our colleagues at the Watershed Conservation Program to carry out a number of studies in the Ohio watershed. We did several years of studies in the navigational pools and the Allegheny and the Ohio rivers, and we filled in information gaps about these deep, mysterious habitats that were pretty understudied until we began tackling them. And we learned a lot more about distributions of rare species like the pistol grip and the club shell. We worked with the Forest Service and studied stretches of the Upper Allegheny River, the Tynesta Creek, and the Clarion River to get a handle on mussels that occur in and around the Allegheny National Forest. In the Erie watershed, we've conducted collaborative projects with Central Michigan University and Buffalo State to study the native mussels in Presque Isle Bay 30 years after the zebra mussels invaded. And we wanted to know if the native mussels have been able to make a comeback after zebra mussels. Next slide, please. So in the French Creek watershed is kind of special to a lot of us. It's a very biodiverse place. It has a number of freshwater mussels and fish. And so we're building on some of the initial efforts by Charles Beer and other members of the Heritage Program conducted basin-wide assessments to determine the population dynamics of some of the freshwater mussels there. And the results really demonstrated that the watershed has some of the most robust populations of mussels in the region. And so we did the long-term work there really highlighted the need for regional conservation efforts in the French Creek watershed. And so areas along the creek and its tributaries have been a real emphasis in our conservation work, and we've conserved more than 5,600 acres in the watershed because we really have a good understanding of how important that place is. So I'll pass the baton back to Eric now.
So now we're going to learn how you do mussel surveys. Doing the survey work is the fun part. Doing all the final reports and everything has to happen, but this is where we really enjoy ourselves when we're out in the field. I prefer doing snorkel surveys like Mary and Danielle are here up at Hunter Station. If you don't like to get wet, you can use a mussel bucket. We can use a 5-gallon bucket where you cut the bottom out and you glue a piece of plexiglass to it, or you can use the really fancy fish and boat or U.S. Fish and Wildlife Service style bucket. If it's deeper than about 3 feet or so, I like to wear scuba because it feels like you're bobbing for apples up and down all day long, trying to stay on the bottom to get the mussels. So that depth is where we kind of trigger and go into scuba, which is a little bit more cumbersome, but you get much better results when you're doing your surveys. If you're going to be in one place for a long time, like we were up at Hunter Station, the consultants like to use surface supplied air. It's called a hookah rig. And you can see there's an air compressor here and a float that's anchored here right in the site. And you can dive 4 people at a time off of this. It's really nice for shallow areas. And you can see these are lines marking our grids here. So these are some of the actual cells that we were salvaging in Hunter Station. Mary had said that, we've done a lot of work in the navigational pools, and that's been some really challenging work. Large bodies of water, large flows. When we're diving in the Ohio in particular, we always wear full face scuba from water quality concerns, but also most importantly would be if you look off the stern of our vessel here, you can see that large barge coming up through pushing typically coals, what they're moving on the Ohio right now. So we always want to stay in communication with the boat so that we know, hey, there's a barge coming up on topside. You guys need to come up for safety reasons. So when we're out doing our surveys, this is what it looks like at the end of a survey. You can see the yellow bags there that I'm holding and Ryan's looking at going, boy, you guys didn't get very much on this survey. You can see the little twist ties on there, marking it off in 10-meter increments. So we'll then be able to take and do GIS analysis and look at where the mussels were distributed throughout the channel. So probably the most important mussel project that I've ever been involved with in my entire career was the Hunter Station Bridge project. So I put this slide in to give everyone a frame of reference as to where we are on the landscape. We're up in the southwestern corner of the Allegheny National Forest. And the Hunter Station Bridge is all the way up here in the top right of this picture here. Pittsburgh's down here in the southwest corner. We're 5 miles downstream from Tionesta, if you're familiar with that region, about 100 miles north of Pittsburgh. So pretty far out in the hinterlands. Was this bridge that was causing some problems. It's causing some problems because it's built upon some of the most dense mussel resources that we have in the state of Pennsylvania. And the bridge was in a state of disrepair and needed to be replaced. This is a federal highways PennDOT project, and the consultation started all the way back in the mid-90s. They needed to try and figure out how to get this bridge out of here. They knew there were mussels present. They didn't know how many, so USGS came in and did some surveys. Well, lo and behold, they found some of the densest populations of mussels in the Allegheny River system right downstream of this bridge and at the base of the bridge. So they kind of figured out how we're going to tackle this, and the wheels spun for 14 years. They couldn't figure out how to handle it. Well, in that time frame, 3 additional species became federally listed while the wheels were spinning, for a grand total of 5 federally listed species now in the footprint of this bridge. The project gained traction when PennDOT said, look, we got to get this taken care of, the bridge is bad. So WPC was invited by EnviroScience, who is a consultant from Ohio, we were tasked with quality control and quality assurance for all of the mussel relocation work and all of the salvage work. It doesn't sound like a lot, but on this project, this was the largest freshwater mussel relocation project in United States history when it began in 2015. There's Mary and I and several others at the base of the bridge right here. You can see our bubbles coming up. That's all you can see. There were about 21 people in the water at any given moment on this project. So lots of divers in the water. This project, like I said, has changed mussel conservation forever in Pennsylvania. PennDOT went above and beyond on this project. We don't hear that a lot. A lot of people talk bad about PennDOT, and this project, they did everything way above what needed to be done. They were only required to protect the state and federally listed species, but PennDOT decided to use this as a model project, and they said they were relocating all species, listed, non-listed, common species. Everybody was being moved. I don't think they realized that we'd find over 136,000 mussels when it was all said and done, but nevertheless, that's what happened. The mussels were moved to 6 different states in the Seneca Nation of Indians, The common species were kept in Pennsylvania, and the federal species went out to different states to try and bolster those populations as part of the endangered species recovery plan that the club shell and northern riffle shell have written for them. The goal of the Endangered Species Act is not to keep listing animals, but it's to try and remove animals from that list by bolstering their populations. And I think the Hunter Station Bridge project really helped out with that. If you remember back a few slides ago, that picture was taken off the top of the bridge. This is what you were looking at. These are all of the grids that were set up in the crane pads and the bridge piers for the new bridge that was going to be built. The old bridge is right here in green. That needed to be ripped down, and it was so big they couldn't rip it down, so they actually dynamited it down and it fell into the river, and then they had to drag it out of the channel. Before they could do that, we had to move all of those mussels, so salvage all the animals that were in the way of that bridge demolition. So if you rewind a year, or actually 2 years ago, we were doing surveys in the Clarion River watershed for hellbenders. And lo and behold, Alicia Trexler found a mussel. She always finds everything that you shouldn't find. She's really good at it. And she's like, wow, look at this. So we found a mussel. So we started talking with the Forest Service and said, hey, let's do some mussel surveys in the Clarion. It was so serendipitous that we did that because we had a base record now that we actually had some mussels in the Clarion. And you'll remember that Mary said in the beginning of the presentation that, you know, in the early 1900s, the Clarion River flowed black like ink, according to Dr. Ortman in 1909. So it was completely wiped out of all of the mussels that were present. But in 2014, we found a couple mussels. So there were some mussels hanging on up in the tributaries that have naturally repopulated. So we did find a couple species. We found 2 species, the creeper and the wavy ray. And you pay attention to these circles here because you can see there's 1 individual here, 8 individuals here, and 2 individuals of another. So 2 species and 10 individuals at Clear Creek State Park. We did our surveys from Clear Creek State Park all the way up to Portland Mills. And the black dots show you that we didn't find anything at all. We put our mussels out in the Clarion River, our test mussels, for a year. And Fish and Boat said, if you guys get 60% survival, you should be able to move any mussels that you'd like to into the Clarion River. Well, lo and behold, we had over 90% survival. So they gave us the green light to go ahead and move mussels back into the Clarion. And these are pictures of us moving mussels. Now, when we started this project, it was a really romantic project for me. I was so excited until we actually had to start moving all these guys because they're really, really heavy. We had bags and bags. Those are 50-pound onion sacks for anyone that works in the restaurant industry, completely packed full of mussels. We were taking them and moving them to the sites. These are those exact spots that we moved them to. And if you remember those circles that before only had 10 individuals at CR1, well, now we have 10 species, over 2,763 individuals. So we completely packed these areas with mussels, and you could put another 15,000 mussels out here, but we put mussels at all of these sites here to try and increase, to bolster the populations in the Clarion River for 2 reasons. One, it's much cheaper to move the mussels to the Clarion River than to have the consultant move them down to the Ohio River in West Virginia or Ohio. And 2, it's nice to keep those mussels in Pennsylvania to try and repopulate the Clarion, because the Clarion will never have a chance to be repopulated because it's still cut off from the main stem Allegheny River by the Piney Dam. So to date, right now, we have over 38,000 mussels in the Clarion River, represented by these species across the top here. So we're really rebuilding the mussel fauna in the Clarion River. It's a very rewarding project. We've done our 5-year monitoring, and we've got around 81% survival now across all of our sites. So the mussels are really doing well, and the Clarion is making an amazing comeback. A lot of our rivers in Western Pennsylvania are seeing an amazing comeback. And that's because of the Clean Water Act, and it's also because of lots of different organizations, typically the small watershed groups, working on acid mine drainage. Without all of that work on acid mine drainage, we wouldn't have the mussels spreading the way that we do now. But to take it one step further, we're now starting to get into hatchery populations, hatchery augmentations in Pennsylvania. I'm so excited that we're doing that. Some of the southern states have been doing this for 30+ years. Pennsylvania just got into the game in 2019. Scott Ray is running the hatchery up in Union City, and he's doing an amazing job. Mussels are some of the hardest taxa to try and propagate in a hatchery. God loves Scott. I don't know how he did it, but he's figuring it out right in the middle of COVID when you had all those restrictions with employees and hiring people and just trying to figure this whole mess out. And not to mention, they have had to build all of this equipment in-house. Scott is doing an amazing job, and I'm really excited to be partnering with them at this new hatchery with this new NIFWIF grant that we have to try and augment their hatchery operations. The coolest part about having a hatchery now is that we can now raise mussels to improve our populations, but we can also raise mussels for our silo projects that Mary and I have been working on that she's going to talk to you about more in depth. The mussels that we used in our silos, we actually collected the females in Pennsylvania and then shipped them down to Kentucky where they infested the fish and reared them down there and then brought them back up. So we'll be able to do that here in Pennsylvania, and then we can propagate, hopefully one day, federally and state listed species here at the hatchery. When you're removing glochidia in the hatchery, it's a lot different when the bass comes down and tries to bite the lure. We just use a hypodermic needle there and pop the gill and then rinse out the glochidia. This is Scott putting some on the Petri dish here underneath the dissecting scope. And you can see in one drop of water here, these are all baby little clams here, little glochidia. The really coolest thing is this is a single grain of table salt. So you can get an idea of how small those glochidia actually are. You take your bass out of their holding tank, put them in the infestation chamber, dump in the glochidia, wait a certain amount of time because you can calculate based on how many glochidia there are in your water. And after about an hour and 15 minutes, if you look at the gills of this largemouth bass, you can see all these little white dots. Those are our glochidia on our fish for propagation. So tons of potential for hatchery mussels here in Pennsylvania. And I think what Mary's going to talk about next should really excite everyone because it really excites me with the future of mussel work in Pennsylvania.
Okay, great. Well, I'm going to finish off the presentation talking about this experiment that we did, and then lead us to the end with some of those actions that you can take. So yeah, as you know, wonderful things began to happen with the hatchery, and we have some experience with relocating mussels. Eric and I and some of our colleagues at the Pennsylvania Fish and Boat Commission started to collaborate on how to prioritize mussel restoration projects that we'd like to do in the future. And so we began asking questions: where should we put them? If you stock mussels in a stream and it's initially successful, you know, is that the end of the line for those mussels? Will there be another generation? And can that next generation of mussels survive? So we wanted to know how the youngest, most sensitive mussels, the juveniles, would do in some of these areas of interest. And so we decided to examine these questions with an experiment. So next slide, please. So our study design was to compare mussel growth in caged mussels in streams with a history of pollution, but that seemed to be recovering, and compare that to streams where there's a relatively intact mussel community. So the goals were to understand whether young mussels could grow well in these recovering streams and compare them to those with rich mussels. So the streams at the bottom of our table there, the Beaver River, the Clarion, the Dunkard, the Kiski, Mahoning River, and South Fork 10 Mile River are all impacted by different types of pollution. And there are either no mussels left there or a select few from, you know, a formerly rich mussel community. And the streams at the top of the table are streams where we think mussels are doing pretty well, just based on recent surveys. Their mussel community is pretty rich. So we set out to put juvenile mussels in all of these streams. Next slide, please. We needed mussels for our experiment. So we collected females of the plain pocketbook. So they have larval mussels on them. And so we collected those and we worked with a hatchery to extract the larval mussels and raise them. And so they grew them out to a size of 3 millimeters that when we used them for our experiment. The plain pocketbook's a fast growing mussel that occurs in the Ohio Basin in Pennsylvania. Next slide, please. So we had the mussels and then we needed the cages. So they're called silos. So we made our own silos with salad bowls as the molds for our concrete cages. And so we poured concrete and we had some PVC pipes in the middle to create kind of a central flow through chamber there where the juveniles were going to go. And we used a kind of a screened piece of PVC to house the mussels in the middle there. Next slide, please. Okay, and you can see our completed silo on the right side there. And the cross section in the middle shows you, how those silos work. The dotted lines are water. The design of the silo encourages water to flow through that central chamber where the mussels are occurring, or where they sit in this cage. Next slide, please. So last summer we started our experiment, and we put our mussels out in June. We put those tiny little mussels, 20 in each of these silos, and we put 4 silos in all of our city streams. And before going into the silos, the mussels were measured and weighed. So we had an idea what their starting points were, and they were set out in the streams for 2.5 months. We really didn't want them to get washed away, or so we tried to find places where we thought the sediment was relatively stable and they wouldn't get filled with silt. And also kept in mind that people like to mess with stuff and tried to put them in places where we didn't think people would disturb them. We were worried about vandalism and so forth. Next slide, please. So we collected the mussels 2.5 months later. And when we picked them up in September, some of them looked quite different. You can see how much they grew. And silos are actually kind of hard to find. You can see how they look underwater. They got some algae on there. They really blended in with the stream bottom. We actually had kind of a hard time finding them again. Next slide, please. So here's just a little bit of the results. Eric and I have been analyzing the data and writing that up this spring. And so these are our streams that we're interested in potentially stocking. And we found that we had pretty good survival overall. And the mussels grew pretty well, or very, very well in a lot of these places. So we think that the majority of these streams could, be places for future stocking projects. Dunkard Creek was our one exception among this group of streams. There was pretty low survival. That system has a lot of water quality issues, so it wasn't too surprising. So we'll use this information for future projects. Next slide, please. Okay, so wrapping up this portion of the presentation, we owe a huge thanks to all of our colleagues who have let us drag them through the mud, all kinds of crazy conditions, to the bottom of the Ohio River next to barges. And just a huge shout out to our crews who've helped us over the years. Next slide, please. And here's a lot of people who have worked on our projects over the years. So big thank you. Next slide, please. All right, so how do we help freshwater mussels? Well, on Earth Day, it's great to think about these sort of ideas. What can we do to protect our streams and the things that occur in them? Next slide. So just one real basic thing we can do is prevent our local stream from being polluted by things that get dumped down drains, our own sinks, and even our stormwater drains in our neighborhood. A lot of people don't realize this connection between drains and the pipes that go right out to the stream. So for our mussels' sake and our other aquatic fauna, properly dispose of your household waste, please. Next slide. And we have properties with stream banks and streams flowing through them. And so for our landowners that have those wonderful assets, we need to respect the riparian habitat, so that area right along the stream is actually contributes greatly to the water quality and the habitat for our freshwater mussels. And so you can benefit the stream habitat actually by not mowing right up to the stream. That vegetation is important for protecting the water quality and shading the stream and other great benefits. So maintain your trees and plant plants along your stream bank. We do have some programs at WPC where landowners can receive trees to plant in their riparian zone. And so I'll give you some more information about that in a couple slides. Next slide, please. We did mention invasive species a couple places in this presentation. And I and some colleagues work on this IMAP invasives tracking database. It's a free online system at paimapinvasives.org. And we maintain distribution information for a bunch of invasive species in Pennsylvania. And so if you find a plant or an animal that's an invasive species, you can record where it's at using our database. So there's a lot more information if you go to the paimapinvasives.org website to find out a bit more about invasive species, the database, how to use it. We also have ongoing webinars, so check that out for some more information. Next slide, please. And lastly, we do have ways that you can directly connect with us. You can assist with those riparian buffer plantings that I mentioned before, or actually request trees for your property. And if you find mussel shells that you'd like to let us know about, you can also contact us. You can help with projects that do water quality monitoring. Our land stewardship program has volunteer days for doing things like trail maintenance or removing invasive species, and they can use help from volunteers as well. Our Gardens and Green Space program also uses volunteers to plant gardens and to have garden stewards who take care of them. Next slide, please. And thank you so much to our members who are attending. Thank you so much for your support. If you'd like to further support WPC, go to waterlandlife.org/donate, and you can help us out at that link. Next slide, please. And thank you so much to everyone who attended the webinar. This was really fun for me to get to talk about mussels to such a big audience. And I think we have a couple of questions in the chat, but if anyone else has any questions, feel free to put them there now. And Carmen will, I'll hand it over to you and you can let us know what people have been asking. So thanks again.
Hey, thanks, Mary. Thanks, Eric. Great presentation. Hi, everyone. I'm Carmen Bray. I'm the Director of Communications for the Conservancy. And thanks, Mary, again, and Eric. What a great presentation. Lot to unpack there. But we do have some questions, and people were asking a few things related to mussels and the difference between a mussel and a clam. Mary, is that something that you can answer?
Sure, I'll take a stab at it. Yeah, it's one of those things where, you know, people, there's the kind of common use of the word clam, and it's a little bit different than how scientists use it. But most people think about clams as being, you know, what you eat, what you get off of a seafood menu. And those are the, you know, marine variety, and their shells are kind of circular in shape, and usually the where they attach to each other, the two shells, you know, attaches at the hinge, and so they're very round. The shells are very round and symmetrical, and that hinge is kind of at the upper and topmost part of that shell. So that's probably what most people think about when they think about clams. We do also have an Asian clam. That's an invasive species. It's a freshwater organism that's been, you know, occurs in a lot of our waterways in Pennsylvania. And it also has that kind of general morphology, the round shape, the centrally located hinge, and they're small and yellow, and they're kind of ribbed. You know, they're probably about the size of a quarter or so.
Awesome. Thanks, Mary. This is a question for Eric. We have a question from Randy who's asking, how do we, or how is the relocation of mussels to other states? How does that impact our streams here in Western Pennsylvania, specifically Northwestern Pennsylvania?
Yeah, that's a good question. The population in the Allegheny River, where Hunter Station is and where those mussels came from, is so robust that according to USGS, they have a site that's actually denser downstream from Hunter Station than what we found. So to give you like an example, a normal mussel stream, a decent mussel stream would be around 2 mussels per meter squared. You agree with that, Mary? Plus or minus. Hunter Station, we had 36 mussels per meter squared, and 32 of those were federally listed species. So extremely dense populations. So there were mussels to spare to go to those other states. That's why that was U.S. Fish and Wildlife's decision to move them to the other states. And they needed to be moved out of the footprint of the bridge. And it just made sense to try and use this project as like a once in a lifetime kind of thing to try and bolster those populations range wide.
Great, thank you. A couple of questions related to, this is another one maybe for you, Eric, for the Clarion River relocations or introductions. Are there any host fish that are, maybe the best that are being used or preferred, especially when it comes to some of the work that we've been doing?
Yeah, that was one of the components. When you do a presentation that's this short, Mary and I could probably talk 3 hours each about what we both did today in 30 minutes. We had to go out and collect all of that data on the host fish species that were present in the Clarion, because you wouldn't want to move mussels there if their host fish isn't present to allow for reproduction. So yeah, there's, I think, 20 or 21 species of fish that are used by a variety of the different mussels in that stretch of the Clarion River, with probably the most common species being your smallmouth bass, some of the river chub, no, not smallmouth bass, I would say, and creek chubs would probably be the most common in that stretch. But that was a huge component to the work, was making sure that the host fish population were present.
Great, thanks. This is a question for either Eric or Mary from Rob. He asks, are there any future plans for surveys or introduction programs in the Youghiogheny River or the Castleman River?
Well, I think, yeah, Eric might have a different take on it, but, I think those places we probably need a bit more survey work. Of the two, I know the most about the Youghiogheny. It's kind of an interesting, it's a large river in that southwest corner of the state, and it flows into the Monongahela, which has, had a long history of pollution. So it might be kind of a, situation where it's, in relatively good condition. It could, I think it's an area where mussels could occur and maybe could thrive. My understanding is they're kind of at perhaps low numbers. So I think we need to do a bit more study there to get some information about that system, but there's some potential, I think.
Yeah, and the Castleman is wonderful in Maryland, but when it gets to Pennsylvania, there's a lot of acid mine drainage issues that lots of groups, like I said, are working on, remediating those issues. But, I think it'd be a great place to put some silos out in the future. If we're able to do some more funding work for doing more silos, I think that would be a great spot.
Another question, does Pennsylvania have a native freshwater clam species?
That goes back to the first question, that the word clam and mussels are sort of used interchangeably sometimes, and it sort of creates some confusion for sure. So the native freshwater mussels that we have also can be called clams too. So it sort of depends on, who's saying what. It's, just the layman's terminology. People can use those two words interchangeably. So the freshwater mussels or clams, their anatomy is similar to some of the marine clams that we have. And so they're sort of in a group that's a little bit, that's separate than those marine mussels. And so anyway, that term is not, it's not a great term, and definitely
No,
creates.
it's not. And you also have the fingernail clams too. So I mean, if you want to,
Yeah, exactly. So we'll spare you from all the
we don't need to go that far.
Nobody wants to hear it.
We have a couple more minutes, but if anyone wants to stay on after we conclude at 1, we're more than welcome to hang on the line to answer some more questions. But let me go through a few more before we officially wrap up. I have a question from Donald who says, how large a stream is necessary to support mussels? And would any of the Blacklick Creek tributaries be suitable with respect to water quality?
Oh, well, I mean, I can maybe take the first one. The smallest stream I've found them in are like, second or third order streams, if you're familiar with that terminology. So not, like the teeniest, tiniest little, creek that you could jump across. Usually not, those streams don't have mussels, but kind of the next size up, maybe, for example, like 10 feet or wider. When you get a larger watershed, you tend to have, they need those particles that flow downstream from the water, upstream watershed. And so that's their food source. And so there needs to be enough of that in the watershed for them to live. Eric, do you know anything about Blacklick Creek? I'm not familiar.
Yeah, Blacklick is probably one of the most beautiful watersheds that I've ever set foot in. Unfortunately, the acid mine drainage in that system is still pretty tough. I think once the acid mine drainage is taken care of, there's the potential for, it's pretty wide down at the confluence with Yellow Creek, or Tulick, I can't remember, I think it's Tulick it flows into. But we have not done any work in that area at all, so I can't. If I were to speculate, I would say no, not yet, but look at where we're at now compared to where we were 100 years ago with the Clarion in particular. There was no fauna at all. And the next 100 years, I mean, the future is so wide open right now, it's crazy. So maybe fingers crossed.
Yes, fingers crossed. Thanks, Eric. This is our final question. This is coming from Meredith. She's doing some research at Duquesne University, and she's wondering if there are any opportunities for people to join our surveys.
Yeah, we could try and make something like that happen. Just you see my emails there, Mary's emails there, send us an email, we'll work something out.
Well, again, thank you, Eric, Mary, also Jennifer, Chrisman, who introduced our webinar today. This webinar session has been recorded. It will be placed on our website, waterlandlife.org, as well as everyone will get a link to say that it is available for viewing within the next 24 hours. We'll have that up on our website. So just want to thank everyone for joining us today. Thank you for your time. Have a wonderful Earth Day. Hope you learned a lot today about freshwater mussels. If you want to stay on the line, feel free. We'll hang on for another 15 minutes or so to answer a few more questions. Wishing everyone a great day, and thanks again for joining us. Okay, so I do see some folks who are fouling out. Some folks are still staying on. So Eric and Mary, you guys are still there? You want to still hang out for another 15 minutes or so?
Sure.
Okay. We do have some questions of when the recording will be available. We did talk about that. Here's a question. So either Mary or Eric, how does the Aquatic River look for mussel placement?
Oh, the Conemaugh.
Conemaugh, sorry. Yes, Conemaugh. Thank you, Mary.
Yeah, well, gosh, I know well, we can maybe tag team on this one. I know I did look for mussels a few years ago and didn't find much there. But I understand that there's been some AMD treatment going on in that watershed. So things might be looking better for it.
Yeah, I think it's going to be in that status of like the Kiski. You know what I mean? There's a chance that now that the water quality is starting to get better, that we could see mussels at some point. And there's going to be some impoundment issues, with the Army Corps facilities that are on there. So natural mussel recolonization is going to be a struggle, but we've overcome that on the Clarion, so and anything's possible.
I have a question from Michelle. We have about 31 people still on the line. And she's asking, what happened with the invasive zebra mussels in Lake Erie? Were they ever a threat to our rivers and creeks?
Yeah, they have spread to our rivers and creeks. So they're in all our major waterways in Pennsylvania now. Really they seem to only take off though in places that are really the slow moving rivers. So we did see them quite a bit in our surveys in the Ohio River. So that was in the navigational pools. So they're essentially, these areas between those big dams on the Ohio River. So the water flows really slowly. It's more like a lake system. And so there throughout that system, parts of the Allegheny navigational pools, they occur. But the faster moving parts of the river, like the upper Allegheny, they don't really, thrive or survive there. But they're still present in Lake Erie and Presque Isle Bay. I mentioned that study that we did there, and the fauna there is, dramatically different. Much fewer species of the native mussels occur there now after zebra mussels came through.
And how long does it take for a freshwater mussel to grow before it starts giving us its filter, service of cleaning, the rivers and streams? What does that process look like from juvenile to, they're starting to do that great work?
Yeah, they're cleaning the whole time because they need to filter feed to survive. So their rates are going to be much smaller when they're, a wee little 5 millimeter juvenile, when they're 130 millimeter adult. But yeah, the ecosystem services that mussel provides starts, once they drop off from their host fish.
Great, thank you. Another question. Do smaller streams such as Little Beaver Creek contain populations of freshwater mussels? And how small can a stream be to sustain mussels?
Well, I mentioned the eastern pearl shell and how that occurs in some of those smaller trout streams. But for the most part, they are, medium-sized watersheds or bigger. So, streams that are probably 10 or 15 feet across is maybe kind of a general rule of thumb. So usually not the teeniest, tiniest little streams. And I'm not familiar myself with Little Beaver Creek, so I can't tell you anything about that one.
Yeah, I don't know anything about the Little Beaver. I've never surveyed it, but based on our silo results with the Beaver River, it's probably going to be an area we'll start looking at here in the next couple of years, hopefully. I was really surprised at the survival that we saw over in the Beaver and the Mahoning River as well.
Okay, we'll take two more questions. Here's a question from Donald. Are dams more of a problem or an asset in protecting from invasives?
Yeah. Yeah, that's an interesting question. Yeah, I mean, I think in some cases they offer some protection from invasive species. In say some of the tributaries in Lake Erie, the dams keep some of the invasive species from moving up the tributaries. Like some of the sea lampreys that occur in Lake Erie, I think are stopped by some of the dams. So, I guess you could call it a benefit, but they certainly are an issue in terms of habitat as well.
Great. I'm getting a lot of questions in the chat and questions and answers from Lyndon, who definitely wants some mussel silos in Tumbled Creek. So he's maybe this is a partner that you guys have worked with in the past, but definitely that's a really great watershed group there. So yes, reach out to us, Lyndon, to Eric or Mary for more information on that. have a question related to water temperatures when it comes to if they impact freshwater mussels. So do water temperatures impact mussels?
Yeah, we've seen how they can impact, especially in the silo studies. The juveniles are susceptible to extremely warm temperatures. Mussels are typically more of a warm water species, so they're not going to be as impacted if you're thinking about it from like a climate change for brook trout, how they're going to be severely impacted with climate change because of them being a cold water species high up in the watershed. But yeah, if things get too warm too long, it will be an issue for mussels for sure.
And how old is a mussel before it can reproduce?
That's a good question. I think it varies on species. Where are you at, Mary? I think 2-3 years. Is that general consensus, or is it? I honestly don't know exactly.
Yeah. Gosh, I'm gonna have to say we'll look that up for you.
Yeah.
I don't know.
I don't want to tell lies because yeah.
Well, great. Listen, guys, we've gone through a lot of the questions. If there's anyone still on the call that you do have, or on the webinar, if you do still have another question, feel free to email Eric or Mary. Their emails are right there on the screen, or at info@paconserve.org. We'll get your questions to them as well. Well, we'll officially wrap up for today. Anything else, Mary and Eric, to end on?
Happy Earth Day.
Thanks for your time. Happy Earth Day. Hope to see you out on the river.
Sounds great. All right. Thanks, everyone. Thanks for joining us. Have a good rest of the day.
Bye.
Bye-bye.